Good afternoon, everyone. I'm Guoxiang Xian from Metabol Profiling Company, Professor Wei Jia's team. It's my great honor to give report here. My topic is a novel metabolite array technology for precision medicine. As we know, metabolomics studies the complete set of low molecular weight metabolites present in cells, tissues, and body fluids. It is an important branch of system biology. Metabolites are regulated by genes and influenced by environment, like our diets, lifestyles, and gut microbiota. The interaction of genes and environment constructs individuals' metabolite phenotype. Usually, metabolomy is considered as the result of genome and proteome. However, there is more complicated regulation mechanisms. On one hand, metabolites provide materials and energy for upstream macromolecules, like amino acids for proteins, nucleotides for genes. On the other hand, metabolites interact with upstream macromolecules to regulate body phenotype. For example, at the DNA level, DNA methylation and histone acylation need the interaction of small molecular metabolites, like SAM, acetyl-CoA. At the transcription level, specific metabolites combine with the riboswitch to regulate initiation and termination of transcription. At the protein level, metabolites can allosterically regulate activity of receptors and transcription factors. Metabolites play much more important roles as we used to recognize. Now, metabolomics is widely used in many fields of biomedicine. It is used for disease diagnosis, prognosis, drug efficiency and toxicity evaluation, and pathological mechanism study as well. The typical clinical application is neonatal genetic metabolic disease screening using dried blood spots. By detecting amino acids, organic acids, and carnitines, one can screen hundreds of neonatal diseases at one time. Metabolomics is also used to select new drug targets, which can be metabolic enzymes or metabolite transporters. Thanks to rapid development of metabolomics, more and more metabolic enzymes are found as potential drug targets. Nowadays, some small molecular targeting IDH or glutaminase or fatty acid synthase have entered the stage of clinical trials. Some of which have been approved, such as AG120. Except for drugs, diets also can be used to regulate metabolite levels and improve prognosis. For example, ketogenic diet, Mediterranean diet, low-calorie diet, intermittent fasting, and diets depriving of certain nutrients, including treating tumors by depriving methionine. Finally, the gut microbiome metabolomics. It can affect host immunity and metabolism by secreting certain metabolites. In fact, gut microflora is a treasure house for potential drug targets. Traditionally, metabolomics is mainly divided into two categories. One is untargeted metabolomics. The other is targeted metabolomics. Most researchers believe untargeted method is suitable for discovery research by marker screening. It can detect as many metabolites as possible. The most used platforms include QTOF and QE platform. The targeted method is believed to be suitable for validation research with no targets, like bile acid profiling, amino acid profiling. It provides quantitative results. We usually use TQS or QTRAP for targeted metabolomics. Although great progress has been made recently in metabolomics, both untargeted and targeted ones, the comprehensive analysis of a global metabolomy in biological specimens has not yet been achieved. The clinical usefulness of application of metabolomics has not yet been realized. There are still great challenges for metabolomics in clinical application. Firstly, the most important problem is the lack of standardization. Untargeted methods generally provide relative concentrations in biofluids or tissues of subjects with disease to determine how they differ from those of controls, but not absolute concentrations. The results obtained from one lab may have significant difference between the other. Secondly, no single analytical platform can cover the entire metabolomy because of the vast chemical diversity and the great concentration range of different metabolites. Like hydrophilic metabolites of amino acids or organic acids, nucleosides, sugars, and hydrophobic metabolites like lipids, long-chain fatty acids. Usually, these metabolites need to be analyzed in several platforms. However, these technologies are generally labor-intensive and lack high throughput capability. Meanwhile, the concentration ranges of metabolites can span over a dozen orders of magnitude, with compounds like glucose in millimolar range in blood, and compounds such as some eicosanoids down in the femtomolar range. Methods suitable for quantitative analysis of hundreds of metabolites in a biological sample are emergently needed for clinical research. This method should have advantages of both targeted and untargeted metabolomics, like covering many kinds of metabolites of untargeted methods and absolute quantification of targeted methods. This method should cover many types of metabolites with absolute quantification ability. Now, I want to simply introduce our company. Our company is located in Shanghai, China. Our company was founded by Professor Wei Jia, who is the director of Phenotype Research Center, Hong Kong Baptist University. He has more than 17 years experience in fields of metabolomics. This is some experiment or equipment of our lab, our company. Our team focuses on the development of metabolomics analysis methods and applying these methods to translational medicine. Here are some representative publications by our team recent years, like include the methods development and application. In addition, our company also developed metabolomic kits for research and clinical use. Back to previous topic, many efforts have been made to develop such a quantitative metabolomics methods. Similar papers have published. Several papers have published. For example, in this paper published by Professor Wu, metabolites with carboxyl groups was derived and detected by MRM. This method can quantify 67 compounds, including amino acids, organic acids, short-chain fatty acids, PUFAs, bile acids, in one injection. And in this paper, metabolites were derived by 3 different reagents and mixed together for detection. And it can quantify polyamides, amino acids, indoles, bile acids, and free fatty acids. In total, there is 80 metabolites can be quantified. Here we report a novel metabolomics technology using automatic chemical derivatization (LCMS) approach that can simultaneously determine over 300 metabolites belonging to different types, which may named Q300 quantitative metabolomics. The metabolites include amino acids, fatty acids, organic acids, bile acids, carbohydrates, benzenoids, carnitines, short-chain fatty acids, indoles, phenylpropanic acids, and other metabolites. Important primary metabolic pathways such as glycolysis, tricarboxylic acid circles, fatty acid oxidation are all included in this method. It is noteworthy that microbial metabolites like bile acid, short-chain fatty acids are all included in this method. All metabolites mentioned were absolutely quantified with stable isotope labeled internal standards. This Q300 quantitative metabolomics has minimum cross-platform variability, improved robustness, and maintaining a global metabolic profile with far more detailed information. Invention patent in China. Invention patent have been approved in China, and the methodology paper of this technique is still being submitted. The Q300 quantitative metabolomics is highly automatic. The sample processing is very simple. The experiments were performed in 96 well plates. Procedures like metabolite extraction, derivation, dilution can be performed in automatic pipette workstation. This workstation, like this. This is a real chromatography spectra of standard mixture, except for calibration standards, QCs, and blanks. 86 samples can be detected in 196 well plate. Here we listed the minimum sample amount needed for different sample types. Because of the high sensitivity, we only need a small amount of samples. We need 20 microliter of serum plasma or urine. 3 pieces of dried spud spots with 3 mm diameter, 5 mg of frozen dried feces, 10 mg of tissue samples, 1 million cells. Here we listed the recovery of some representative compounds, like branched fatty acid leucine. The recovery rate is 102. The important fatty acid DHA, HPA, ARA, or the recovery is 93, 110, 102 respectively. And bioacids and some short-chain fatty acids. The recovery are all between 80% to 120%, means the high accuracy of this method. There are over 300 metabolites measured in one injection. When thousands of samples analyzed in clinical study, it is almost impossible to quantify these metabolites with commercially available software like Target Links or Mass Hunter. So we developed our own quantification software, TMBQ, the Targeted Metabolomics Branch Quantification Software. It is the first cross-vendor platform for LC-MS-based high-throughput metabolite quantification suitable for data generated from different LC-MS systems, such as Waters, Agilent, and ABSEX. There are 7 functional modules in the main workflow of TMBQ, including data upload, smooth and denoising, peak identification, peak intensity calculation and IS correction, standard curve fitting, concentration calculation, result exportation. This is the main results exported. We provide all the results include concentrations and chromatography spectrum of each metabolite in every sample. We also provide all standard curves of every metabolite. Results of Target Links and TMBQ were systematically compared. The Pearson correlation coefficients of the same metabolites quantified by TMBQ and Target Links are all higher than 0.96. It means the result of TMBQ is comparable to that of Target Links, and TMBQ is much more fast and effective. And also, this software is the patent of this software is approved in China. This is the patent number. The assay kit was used to probe the changes in metabolic patterns in chronic liver disease patients. The sample set includes 342 healthy controls, 349 HBV patients with fibrosis, and 543 liver cancer patients. Over 1,000 samples were detected in this study in total. 17 assay kits were used for the analysis of all the samples. 5 pooled QC samples were analyzed within each kit. A multivariate control chart was established using the score value of one model dimension of PCA, this finger. MCC displays the data measured on the process over time and monitor variations produced by a series of laboratory procedures. As shown in this figure, the score value were all within one standard deviation, which suggests that the data were robust and suitable for further analysis. Here the yellow spots is QC sample and the black spots is every clinical sample. The PCA mode of all the samples shows a clear separation of the three groups. The blue is control sample, the red is HBV with fibrosis, and the black is liver cancer patients. The results show that metabolites concentration was significantly altered among control, liver fibrosis, and liver cancer patients. Notably, 12 metabolites like aspartic acid, cerionic acids are abundantly in control and were gradually decreased in fibrosis and cancer patients. 12 metabolites like L-tyrosine glycolic acid were increased in liver disease patients when compared to healthy control. These altered metabolites were validated in independent cohorts, and finally 5 metabolites were selected as further diagnostic marker for liver fibrosis. These markers were used to develop diagnostic kits of liver fibrosis in our company now. In a word, our method, with this method, we find a powerful way from metabolomic research to clinical diagnosis. Many other sample types were measured by this Q300 method, like feces, urines, liver and brain tissues, dried blood spots. Usually over 200 metabolites were accurately quantified in each sample. Over 30,000 samples were measured by this method until now. Biological samples from COVID-19 patients have also been detected by our clients using this Q300 method. Now we have developed Q300 kits. Researchers can perform the experiment by themselves with our kits, only if they have UPLC triple quadruple MS system. TMBQ account will also be provided for metabolite quantification. Our kits are suitable for all mainstream triple quadruple MS systems like Waters, Abasex, Thermo Fisher, Agilent, and so on. We also provide IMAP cloud platform for subsequent omics analysis. Our cloud platform includes common omics analysis like multi-dimensional PCA, PLSDA, OPSDA, and using single-dimensional statistical tests such as t-test, u-test, variance analysis, and pathway analysis, including enrichment analysis, topological analysis, and diagnostic analysis. And this IMAP soft platform includes Chinese and English version. In conclusion, Q3 kits is a good method for large-scale clinical research. It's high throughput, accurate, fast, and good compatibility. Sample Processing is highly automatic. Over 300 metabolites can be quantified in one injection. Every metabolite has its own standards and isotope labeled internal standards, which guarantees high accuracy. TMBQ makes the fast quantification, and this platform is suitable for all mainstream instruments. In the future, data generated from such standardized procedure can be used to accurately translate metabolomics outcomes into areas of clinical research. Ultimately, the development of a clinical metabolomics database will facilitate the use of metabolomics data in personalized medicine. In fact, the upgraded version Q600 kit is under developing. Except for all the metabolites in Q300, Q600 can detect over 300 lipids. In 2017, we report an automatic quantitative analysis method for microbiome metabolite based on GC12 for the first time. Now we have transferred this method to UPLCMS platform and improved. This is a representative chromatography spectrum of Q600. Finally, I want to show our service product. Except for Q300, we also provide regular untargeted metabolomics and lipidomics analysis, and absolute quantitative method for dozens of different metabolites, like bioacids. Our bioacid detection method can quantify 65 bioacids in one injection within 15 minutes of each analysis, and amino acids profiling, organic acids profiling, fatty acids profiling, and stereo hormone profiling, nucleotides, coenzymes, vitamins, and neurotransmitters. In addition, we also provide microbiome sequencing and isotope flux analysis. This is our WeChat account. You can scan this and follow it. So far, we have cooperated with many national and foreign clients. Here are some of their publications, like Cell Metabolism published this year, Immunity Guts in 2019, Advanced Assay, Hepatology, Signal Transduction and Targeted Therapy, and so on. That's all. Thank you very much.